Welcome back to biology. We're going to be doing the last half of the Investigating Biological Compounds lab. We've already done the pre-lab, which is essentially part 1, the starch test, and we're going to jump into part 2. Just a recap, you need to make sure that you've read through the background information in the pre-lab and tutorial sheet. There are right there questions that are referencing the information in here. So if you're going to come and ask me, Mrs. McAnally, where do I find out what an indicator is? I'm going to tell you to go read. It's right there. If you're going to ask me how do I answer 2, 3, and 4, it's right there. So find out by reading this part here, and then if you still have questions, you can come and see me. Procedures wise, so this is how to open it, what to print off, view the tutorial. You should have already have all this done when you came into class today. We did part 1 together. There's a video on that if you need to review. We're starting here at part 2, test for proteins. Okay, let's get started. We're going to go to begin the lab. Choose your gloves. Okay. So, as you can see from the setup, just to recap what we're looking at, we've got all of the solution samples and the indicator solutions that we need up here on the top shelf. So vegetable oil for lipid, water, milk, starch for carbohydrate, and the indicator for that, which is iodine, sodium hydroxide, and copper sulfate, are the indicator for the protein. Sudan 3 is the indicator for the lipid, and then you have albumin and unknown. So I know we discussed this, but I'm going to just remind you that albumin is a fancy word for egg whites, and egg whites without the yolks only have protein. So this is what we're using to test for protein. This is what we're using to test for lipid. The starch is what we're using for carbohydrate. Now remember, starch comes from plant carbohydrates. That's what the plants store. And then the unknown, I'm not going to tell you what that is because you have to figure that out after you've learned what they should look like. And then we're also going to test milk, which is going to be test 5. I have already set up what we already had done, which was the test for starch indicators. So we can zoom in on that just briefly in case you didn't get a chance to see that close up. So this reminds us that if you put iodine in water, you're going to get diluted iodine. So you're not going to see a color change, just a lighter color. And if you put it into a starch, you should see a really pretty purple color show up. And I like to compare it against the white of the wall. So if you see the color change from brown to purple, then you'll know that a carbohydrate is present, in this case specifically starch. So we're starting at part 2, which is where we're testing for proteins. And that's these 2 sets of test tubes. The third part is the test for lipids. And that'll be these 2. The fourth part is the test for the unknowns. That's these 3. And the 5th part is the test for milk, and the last set of test tubes. So what I'm going to go ahead and do for the sake of time, since if you notice in your procedure, it says to add a pipette full of water in the test tube labeled protein control, and down here in part 3, lipid control. So just like we did for starch, the first test tube, the control, is going to be water. So I'm going to pause the video and set these 2 up so that all I have to do is add in the indicators. So I'm going to put water in the first, then the sample in the second, and then I'm just going to add the solutions, the indicator solutions, to each of the test tubes. So I've already got my water in my part 2 and part 3 test tubes. At this point, I've got to get the sample for the protein, and remember we said that's going to be albumin. It's already got its own dropper. So we're going to take some egg whites and we're going to put it in the test tube. And put that back. And then for the lipid, we're going to use the lipid dropper. And if you can, remind me why. Think about this. We're going to talk about it in class again. But the reason that we have in specific droppers for specific of these three that don't already have their own droppers, oil, water, and milk, is that we don't want to cross-contaminate. If I use the same dropper for the oil and then go to the water, not only am I going to have water in my oil, but I'm going to have oil in my water, which I use for all of my controls, and that's going to really mess up our data. So let's go back to part 2. It says place a pipette full of vegetable oil and then test for the lipids. Now what do we do to test for the, that's for part 3. We test for the proteins. So for the proteins, we need to take these 2 indicators and put them in both of the test tubes. So 1 squirt of each of these and 1 squirt into of the other. This for proteins requires 2 indicators to work. So keep an eye on the protein test tubes when I put the second indicator in. Oh, come on, don't do this. All right. So, sorry, I should zoom out. But you saw a pretty significant color change. We've got to get our stirring rods, a clean one every time, because you don't want to cross-contaminate. And then we can look at this closer up. All right, so this was the original, and this is what happens if a protein is present in our sample. And you can tell that because even though this is blue, if you look at the original indicators, the 2 combination indicators, one of them is slightly blue, so that makes sense. Between the water and the copper sulfate, it's going to be a little bit blue in the test tube. But what you're looking for is a significant color change from light blue or the copper sulfate diluted to black. Right? If there's a protein present, the solution will turn black. So let's add the lipid. I've already got water in it. We already added our lipid in it. And here's where you have to be careful. Some of us don't have really great vision for color. There, no derogatory statement here. It's just a fact. But what we're looking for is a change in color family. The Sudan 3 is very clearly orange. So you can tell that if you zoom in, the Sudan 3 shows up pretty distinctly orange. So what we're looking to see is any change away from the color of orange. So it's going to go toward a pinkish color or a reddish color if it's present. And if you look at these 2 and you compare them, compare them to the white, and you can compare them to the Sudan itself. Okay, you can tell that the water solution was just the Sudan diluted. This one is different in color. It is hard to tell, but it is more reddish. All right, so if a lipid is present, it's going to turn this color, more of a brighter red color. I will take a moment to caution you that when we use milk, you have to take into account that milk is already white. So whatever the final color will be, you have to account for the fact that milk was white to begin with. You're just looking for a color change toward a red family. All right, so on our data tables then, we've gotten our first 3 parts filled out. Going back to our lab report, make sure you have this opened already, and I showed you what this looked like on Friday. Your data table is required to be filled out. I've put this row up here at the top. You don't have to fill the water row out, but if you did, if you put iodine in water, you'd see lighter brown. So, but you wouldn't see a color change. You wouldn't see a color change across any of these. Just a dilution. But for the starch, if you remember, and you can see it here in the background, it went from brown to purple. So brown to purple. Purple. For the protein, it went from clear to black. And then for the lipid, it went from orange to reddish. Okay. And now we're ready to do the unknown in the milk. And then you'll fill in those data points too. So let's slide down here to the unknown. Now notice here down in the unknown, I don't need to put any water in the unknown or the milk test tubes because I've already got my samples for water for my controls, right? But what I need to do is put the same substance in each of these test tubes and then put the iodine, the sodium and copper combination, and the Sudan 3. So let's do that. Let's add our unknowns first. 1, 2, 3. Now we're going to test for starch, and we need iodine to test for starch. We're going to use these two to test for protein. And for lipid, we're testing with Sudan. So when you look closer, all you need to be doing on your data table is filling out whether or not there was a color change, which would mean there's a starch present in our unknown, whether or not there's a protein present, and whether or not there's a lipid present. And we would do that by looking at the colors and the potential changes. So I think it's pretty clear to say that there was a starch present at some point in our unknown because it went from clear to purple. And then for the protein, it didn't change from the unknown color when I added a protein in it. So do you see that there's no color change to black? So there's no protein. And although it's a little bit trickier, for the lipid, we can tell that this color is closer to the water than to the indicator for lipid. So there is no lipid in our sample. And we can tell that because it's not the same color. It is closer to the water than the positive indicator for lipid. It's easier to see when you put them close. I know that's a little harder to tell, but there it is. So on your data table, you would write in color change. So you'd write purple. You would write no change. And you would write no change. That's what you would do. Okay, for the milk, it gets a little bit trickier because remember, like I said, the milk is white to start with, which you might not realize, but when you add a white substance, that's going to have an indication. It's going to actually be easier to see the color changes. So let's use the milk dropper. I'm going to go over here. 1, 2, 3. And I'm going to use those same 3 indicators. I would check for starch. I will check for proteins by adding both of these. Come here. Ooh. And I would check for fats. Now that we're done with the 5th part, you should be able to see that you don't have a color change from brown to purple. So in the starch test tube, we're testing for starch. Remember, starch is a plant-based carbohydrate. I'm not saying that there aren't sugars in milk. I'm saying there's no starch in milk because starch is how plants store carbohydrates. And that makes sense because milk comes from cows, if that helps. There is protein. You can tell that because it went from clear to black. And in the milk, the point is that it went from an orange-ish to a red-ish. And you can tell the difference if you compare here. So we're just looking for any indication that moves toward the family of red. For part 5 on your data table, like I said, you're going to indicate that there wasn't a change. It did not go from brown to purple, so no starch. It did go from white to black, and remember, clear to black was an indication for presence of protein. And then it went from white to pink, so reddish was the family, so it did indicate for the presence of fat or lipid. For questions 1, 2, and 3, you're basically using your data table to back up as evidence with a complete sentence to answer why we can say that these 3 indicator solutions, the Sudan, the sodium hydroxide and sodium hydroxide and copper sulfate, and the iodine were good indicators for each of their specific molecules. For 4, it's referencing part 4. Part 4, the unknown. Which test tube showed a completely positive response in a complete sentence and back it with evidence. 5, which test tubes showed a positive response with evidence from your data table. Number 6, you need to explain that you understand the role of the water in each of the test tubes. Here's a hint. The role of the water is actually like right there on the label, or where the water test tubes were. For 7, it's breaking this question down. Describe a chemical indicator, which was our, I think, second lesson target for the day. What is it? What is it used for? And how does it work? So you need to complete sentences. For 8, notice that's 8 points. 8 points. So for 8 points, I want this as a paragraph. I've broken it into 2 parts so you know you make sure you answer both parts. I want you to talk about the unknown and the milk. So it is a 2-part paragraph, but make sure that you do it in a paragraph form, that it is complete sentences, and that you've used evidence and data from your lab. So you can do this. If you have questions at any point, you can ask me, you can watch this video again, shoot me an email or a text. Okay? Good luck. Have fun. If you're done and you want to tinker with dump everything together, see what happens. You're not going to blow yourself up in this lab. Take care, have a good day, and I will see you tomorrow.